Hello students, welcome to EPG Pathshala. I am Dr. Nirupama Trehinpati from Institute of Liver and Biliary Sciences. Today we are going to discuss the molecular mechanisms of pathogenesis. This is from paper Molecular Therapeutics. To teach these students basic molecular mechanisms in pathological processes due to infectious microorganisms, it is important that we should understand this module. This module will help students to know that what diseases generally how it happens and how it differ in their presentation, but are always preceded by infection and colonization of pathogens. This module will also teach them that how different pathogens follow almost similar integrated molecular mechanisms. I am introducing to the different pathogens. How the different pathogens like the facultative pathogens, obligate pathogens, they recognize and infect the host. How these pathogens are colonizing and then they disturbed the immunity. Now under immunity it comes the innate and the adaptive immunity. Through the adaptive and innate immunity modulations, these pathogens make the severity of diseases. Over the past few decades, different pathogens have been scrutinized microscopically, and information about their molecular mechanisms contributed to the understanding of infection and disease. Opportunistic, facultative and obligate pathogens. There are different pathogens. Opportunistic pathogens infect in wide range of living hosts and had the low virulence. Obligate pathogens have the capable of infecting within a narrow host range, can infect healthy as well as immunocompetent individuals. Another class of pathogens are facultative pathogens. They can infect with a narrow host range, however have the capable of surviving outside the host environment too. Cross-kingdom host jump. We need to understand what is the cross-kingdom host jump. Pathogens are diverse and they can infect the different organisms. Disruption of normal physiology of a multicellular organism on infection with pathogenic microorganisms. Few are pathogens. They jump from one kingdom to another, from one taxonomic kingdom to another taxonomic kingdom. Few pathogens occur in plants or animals and then they jumped to different kingdom. Cross kingdom in Aerobacterium tumeficus is globally dispersed soil-borne pathogen. It's initially characterized in plants and induces tumor formation. This pathogen induces tumors by transfer of a segment of a tumor-inducing plasmid DNA to the genome of plant cells, deliver DNA into plant genome. This is the one kind of pathogen which can induce the tumor in plants. It does not only transfer to plant genome, however it does in fungal and even in human genomes. After the integration of this pathogen, there is a formation of tumor in fungus and the human kingdom too. Therefore, this pathogen evolved as a pathogen able to infect different organisms as plants, fungi and humans. Now I will tell you about the common processes in these different pathogens and organisms which they follow for the pathogenicity and infectivity. Molecular mechanisms in different organisms are generally common, which are microbial encounter with the entry into the host, microbial growth after entry, avoidance of innate host defense, tissue invasion and tropism, tissue damage and transmission into the new host. Hosts are also jumping in different kingdoms, from one kingdom to another kingdom. We should understand what is the pathogenicity and how it works. Pathogenicity is the ability of the pathogen to infect and it's called as pathogenicity. Pathogenicity is calculated by their virulence, a relative and quantitative degree of pathogenicity. Now I want to tell you how we can define the virulence of pathogen. It all depends upon how much virulent is a microorganism. But how we will define this? Virulent strength contribute to the colonization. This is the simple presence of potentially pathogenic microbes in or on a host. This depends upon infection, attachment and growth of pathogens, and avoidance of host defense, and pathogenesis leading to mortality and morbidity. This all factors contribute to the virulence of pathogens. Few pathogens strive to attach not only to plant cells but also to human cells, leading to an oxidative burst that result in death of these cells, which can also go to the apoptosis and the complete paralysis of immune system. How microorganism access to the host? A microorganism gain access to host either two main types of routes: mucosal surfaces within the body, which are lining of the respiratory, digestive, reproductive or urinary tracts, or through skin, or epithelial surfaces on the outside of the body, like areas of the skin that are either damaged or compromised due to insect bites, either cuts, scrapes or wounds. Microorganisms can survive in a broad range of environments, such as in the soil, water, mouth, salivary glands, and alimentary tracts of larger animals. Transmission of microorganism is also due to pathogen presence and spreading in the air, which is called as airborne transmission. Another way of microorganism in contact by coughing or sneezing on another person. Contamination via intravenous drugs or contamination through infectious blood transfusions, like in the case of hepatitis B, hepatitis C and HIV. These are transmitted through the contaminated blood. General blueprint of microbial infection. It starts with this, that after attachment of pathogenic microorganism, pathogen may enter host tissue through active penetration process or through wounds or natural openings. Subsequently, pathogenic microorganisms colonize the host tissues. This colonization occurs either intracellularly or intercellularly or extracellularly. In the next stage, progeny of the pathogen is released and dispersed, after which new infection can take place. How the pathogen adhere to the host surfaces? It is important to understand that how some bacteria can enter and proliferate inside host cells, and finally how pathogens may cross epithelial or endothelial host barriers and get across to internal tissues, leading to severe diseases in humans. Despite the different defense mechanism, the host pathogens adhere to the host and then enter to the host. Most of the pathogens enter through wide surface area of approximately 300 to 400 square meters of respiratory, digestive and urogenital mucosa, and thus constitute major sites of contact with bacteria. Intestinal mucus layer plays a key role in limiting invasion by commensal bacteria of the microflora, as mucus is mainly composed of glycoproteins called mucins, digestive enzymes, antimicrobial peptides and immunoglobulins. Therefore, bacteria are often found at the top of this intestinal mucus layer where they interact with mucins, whereas the inner layer of mucus where the concentration of antimicrobial compound is high is normally devoid of bacteria. We talk about specific molecule for the adhesion to the host. They express a variety of specific adhesion on their cell surface. Hosts have specialized structure for interact on. Bacterial pathogens specifically express a variety of specific adhesins on their cell surface, which allows them to interact with the receptors on host cells. Microorganisms also have specialized structure through that they interact with the host, as in case of gram-negative bacteria. They have a diverse array of pili that mediate microbe and host pathogen interactions, which are important in development of disease. These bacteria express adhesion at the tip of the long fibril structure, pili, that extends from the bacterial cell surface. Some pili are also involved in cell aggregation, biofilm formation, DNA uptake, phase transduction, and gliding motility. Pili have also their role in triggering signals in both the bacterium and the host upon attachment, and play an important role in molecular mechanisms of bacterial diseases after the adherence and after infection. As we discussed already, that there are different ways of pathogen entry into the host. Through the adhesion, they infect, and they have the different mechanisms how they enter. Most invading bacteria enter the host cell by using either a triggered or a zippered mechanisms. Bacteria may then replicate in their cytoplasmic or vacuolar niches. How the toxins produced by the pathogens and how these toxins affect. Toxins secreted by microorganisms are harmful. They damage the host tissues by making pores in the cell wall. The repertoire of the bacterial cytolytic pore forming proteins are called as the PTFs toxins. These called as a pore forming protein toxins are produced by both gram-positive and gram-negative bacteria. Pathogen also gain access through the plasma membrane to retrograde transport through the Golgi complex and the endoplasmic reticulum within the host cell. What are the defense mechanism against pathogen, or all the microorganisms? They have the defense mechanism against them. This is that first control of pathogen invasion is the commensal bacteria, which is present in our gut, and these are called commensal bacteria microbiota. These commensal bacteria, they secrete inhibitory metabolites such as acetate or butyrate. In addition to these molecules with antimicrobial activities, shedding of mucus can be another mechanism to prevent. Bacterial adhesion to epithelial surface, as reported in case of gastric mucus colonized by the pathogenic bacterium Helicobacter pylori. However, despite the protection provided by the microbiota, some enteric pathogens are able to efficiently colonize the gut and cross the intestinal barrier and cause serious diseases. Various microorganisms utilize specialized surface-associated adherence factors or adhesins to attach themselves firmly to the host. Despite the presence of multiple host defense mechanisms, bacterial pathogens evolved a wide range of strategies to colonize and invade human subjects. After the adhesion, how the pathogen are recognized by the host. There are various microorganisms utilized specialized surface-associated adherence factors or adhesin to attach themselves firmly to their host. However, the first step is primarily oriented towards outer surfaces of host and involves the adherence of the microorganism to host cells or tissues. By combination of innate and adaptive processes, they establish during the host pathogen interactions. Like in the various hosts, they recognized by the pathogen through the PAMPs. Those are the pathogen associated molecular patterns. Pathogens, these PAMPs are present on the host cells like toll-like receptors, or sometimes it's present on the intracellularly, and they called as endocytic pathogen receptors in the endocytes of the cells. Pathogen regulate the host immune system after adhering and infecting. Close interaction of pathogen and host might trigger infiltration and activation of immune cells and eventually lead to the phagocytosis. There is difference in regular infection to severe pathogenicity. It changes the host innate and acquired or adaptive immune system after the pathogen interaction and the infection. How the host damage after the infection? To develop the severity of disease, pathogen must be able to suppress the host immune responses. As I already told you, it involves the innate and adaptive immune responses. All kingdom, including the invertebrates and vertebrates and plants, rely completely on the innate immunity for their self-defense. Plants also have more or less rigid vascular system that transports dissolved compounds in an aqueous solution throughout the plant. Similarly, invertebrates carry hemolymph, which can flow freely within the body open cavity, making direct contact with all tissues and organs. Like blood of human, hemolymph also contains antimicrobial proteins and peptides. They contain circulating cells like plasmatocytes, granulocytes, which are capable of phagocytosis and encapsulation of invading microorganisms. How they activate the innate immunity? To establish infection, pathogen need to defeat the action of non-specific immune system machinery. They generally, when they defeat the one innate immunity, they activate the others. So, they activate the complement system, which is activated immediately after activating the innate immunity. The lectins tags microbes for destruction by phagocytic cells, causes microbial lysis and generate pro-inflammatory responses. However, many bacteria develop strategy to develop even the complement system. Specific and unique host defense peptides are present and they are blocked by pathogens. Pathogenic bacteria evolve resistance to these host defense peptides which are present on the host, and these pathogens alter the host cell apoptosis through these host defense peptides, and blocking these host defense peptides, they have the host cell apoptosis. There are other several compounds which are present on the host and the pathogens block them, and then that's the reason the pathogens survive, but the host innate immunity is totally blocked. They do activate the adaptive immunity also. Vertebrate system contain unique and efficient system called as adaptive immune system, which contains T cells, B cells, NK cells and NKT cells, which really outlined with the diversity of antigen specific receptors. As I explained you earlier that invertebrates and plants have only self defense innate immune system. However, the human have the innate and adaptive immune system both. Despite the absence of an adaptive immune system in invertebrates and plants, the innate immune system in those organisms still possesses some degree of specificity. Activated adaptive immunity can result into pathogen elimination. However, being in different host defense system have a high degree of fundamental similarities, as we discussed earlier, that the all microorganism mechanisms are similar despite that they are present in the different organisms. Another strategy is to deal with host defenses, suppression of host defense, like with the immunosuppressive effects provided by the immune system, such as downregulation of tumor necrosis factor alpha, a signaling molecule in the human inflammatory response. These are common. With all the suppression of the immune system, the host have the defense system that they can invade beautifully and infect and colonize the host pathogen. Over the last several years, molecular and genetic strategies have been developed to identify genetic determinants of bacterial pathogenicity, especially bacterial virulence determinants. Genetic determinants of bacterial pathogenicity are important to identify which pathogen and what will be the virulence of that pathogen. Especially for opportunistic and facultative pathogens, pathogenicity mechanism in alternative host or in the environment, it is important to identify the pathogen. These approaches, combined with rapid advancing genome technologies, and have allowed investigators to identify the infectious gene network, virulence genes, and also the few pathogenic genes which determines the metabolism of pathogens. Gene loss can also play a significant role in determining bacterial pathogenicity and should not be disregarded. Interactions between microorganisms may lead to the development of pathogenicity towards other organisms. Especially for opportunistic and facultative pathogens, pathogenicity mechanism may be selected for an alternative host or in the environment. There are different environments like biotic and abiotic environment has direct and indirect effects on the success of host pathogen interactions. Interactions between microorganisms may lead to development of pathogenicity towards other organisms too. Molecular and genetic strategy to identify the pathogen determinants have evolved many infectious genes, many virulent genes, and pathogens metabolic genes. These genes we can get it through the sequencing program also, and then in the laboratory the investigators can do the simple RT-PCR and confirms those genes for the bacterial, fungal or any pathogens. So students, let us summarize what we have learned in this module. Different pathogens, which includes virus, bacteria, fungi, evade host defense with similar molecular mechanisms. These pathogens jump into different kingdoms, from soil to plants, plants to animals, and soil to human beings also. Pathogen virulence depend upon their colonization and infectivity ability. These pathogens further regulate host immunity by toxins, which is innate and adaptive immunity also. After, as I told you, that plants and the animals or invertebrates, they don't have adaptive immunity. Only the vertebrates have the adaptive immunity. Despite that no presence of adaptive immunity, still innate immunity have the antigen specific receptors. So pathogens recognize that and destroy them and produce the toxins, which also blocks these receptors and then activates the apoptosis of the cells and cell death. After activating the innate and adaptive immunity, which is important, that how we identify the pathogenicity or how we identify the different pathogens, because these pathogens also change from time to time and their genome also change. So it is important to identify the pathogens, and we can identify these pathogens by through the molecular tools, which are like the next generation sequencing, which are the people are using, investigators are using, and then they are validating those genes by common RT-PCR methods.